Yes, sir.
We are in protein synthesis, you know that. Yesterday we completed protein synthesis in science lab. Now we are going to explain it here on smart board. You all are now aware that proteins are formed from amino
Amino
acids
acids.
in cytoplasm.
Yes, sir.
But under a specialized control of DNA. And you all know that the DNA is present in the nucleus
Yes, sir.
and the information goes from DNA to RNA. DNA to
RNA.
That process is called
Transcription.
transcription. Transcription.
Transcription.
And the message, the information
From RNA.
on mRNA is called as genetic code.
Code.
What is genetic code?
Sequence
Sequence
of
of
logs.
on mRNA. That means according to this genetic code, protein has to be formed.
Yes, sir.
You all know that proteins are having important functions in our cell system. Without proteins, life is impossible. Cell is impossible.
Yes, sir.
There will be no structural components formed in the cell. There will be no functional integrity in the cell without the proteins. So proteins are very, very important for structure and function of the cell. And you also know that proteins are formed in factories called as
Ribosomes.
तो ऐसी information कहाँ से from DNA to RNA it is called transcription.
Transcription.
And the information from RNA to protein is called as
Translation.
ये हमने कंप्लीट कर दिया है उधर और आज हम कोशिश करेंगे फिर से एक बार उसको समझने की.
What nutrients we get from our diet? Our bodies cannot use the food as it is when it enters our digestive system. The process of chemical digestion uses different proteins and enzymes to break down the food particles into usable nutrients our cells can absorb. And where are the instructions to manufacture these and all the different types of proteins we need to stay alive? The instructions to make proteins are contained in our DNA. DNA contains genes. A gene is a continuous string of nucleotides.
Gene.
Yes, sir.
Do you know gene?
Yes, sir.
What is gene?
Segment of
Any
DNA.
segment of
DNA.
Gene controls character
Yes, sir.
of organism. How it controls the character of organism? Through formation of protein.
Protein. Yes, sir.
यही gene बनाए protein. कहा था
Yes, sir,
आया मैं
sir.
कहा
Yes, sir.
protein will act as an enzyme यात enzyme पद के तंग हिसाब से reaction
Yes, sir.
तंग हिसाब character पे. Have you got it?
Yes, sir.
तंग हिसाब character पे. यात नहीं ये protein इस कुछ structural component है. तो ये समझ लो.
Yes, sir.
तंग हिसाब character पे. For example, gene for eye color will code for a protein which will act in formation of this eye color.
Yes, sir.
Kacha samajh
Yes, sir.
So genes function, genes act in the cell system through formation of proteins. Gene छिन पाने काम मतलब सोधी नेबर नेरन
क्या?
यम छि काम करन as proteins.
Proteins.
So protein synthesis is very important for life.
Containing a region that codes for an RNA molecule. This region begins with a promoter and ends in a terminator. Genes also contain regulatory sequences that can be found near the promoter or at.
Have you got the promoter region?
Yes, sir.
It should be transcription aspect.
Yes, sir.
Kacha samajh?
Yes, sir.
Promoter the
Terminator.
Never skip it. Pause.
Sequences that can be found near the promoter or at a more distant location. For some genes,
Promoter terminator also.
Yes, sir.
Actually, the unit transcription.
Transcription unit.
the encoded RNA is used to synthesize a protein in a process called gene expression. For these genes, expression can be divided into two processes, transcription and translation.
Yes, sir.
Gene expression.
In eukaryotic cells, transcription occurs in the nucleus, where DNA is used as a template to make messenger RNA. Then in translation, which occurs in the cytoplasm of the cell, the information contained in the messenger RNA is used to make a polypeptide. During transcription, the DNA in the gene is used as a template to make a messenger RNA strand with the help of the enzyme RNA polymerase. This process occurs in three stages: initiation, elongation, and termination. During initiation, the promoter region of the gene functions as a recognition site for RNA polymerase to bind. This is where the majority of gene expression is controlled, by either permitting or blocking access to this site by the RNA polymerase. Binding causes the DNA double helix to unwind and open. Then during elongation, the RNA polymerase slides along the template DNA.
Sure. You can see open can see in the chain. By the
Yes, sir.
really having part after transcription unit.
Strand. As the complement
This is done by RNA polymerase. RNA
Polymerase.
Polymerase मतलब you can polymerize jama ribonucleotide like नहीं है
Yes, sir.
which
Memory base.
jama adventure. चलो.
is pair up. The RNA polymerase links nucleotides to the 3' end of the growing RNA molecule. Once the RNA polymerase reaches the terminator portion of the gene, the messenger RNA transcript is complete, and the RNA polymerase, the DNA strand, and the messenger RNA transcript dissociate from each other.
Dissociate.
Yes, sir.
The strand of messenger RNA that is made during transcription includes regions called exons that code for a protein, and non-coding sections called introns. In order for the messenger RNA to be used in translation, the non-coding introns need to be removed, and modifications such as a 5' cap and a
Introns के exon छासना DNA introns की non-coding का
Yes, sir.
वन कोड करन कर प्रोटीन बनाए नहीं तो हम छासन exons coding region.
Yes, sir.
Why guys have processing you introns in never cut.
Yes, sir.
Remove
Remove.
was complete was in exons you. Because समझियो
Yes,
so
sir.
like as guess energy in zai
Yeah.
clear.
Yes, sir.
Introns and exons.
3' poly A tail are added. This process is called intron splicing and is performed by a complex made up of proteins and RNA called a spliceosome.
Yes, sir.
Lysosome.
This complex removes the intron segments and joins the adjacent exons to produce a mature messenger RNA strand that can leave the nucleus through a nuclear pore and enter the cytoplasm to begin translation. How is the information in the mature messenger RNA strand translated into a protein? The nitrogenous bases are grouped into 3 letter codes called codons.
Yes, sir.
The genetic code includes 64
Yes, sir.
codons. Most codons code for specific amino acids. There are 4 special codons, 1 that codes for start and 3 that code for stop.
AUG, eh? How was something?
Yes, sir.
Translation begins with the messenger RNA strand binding to the small ribosomal subunit upstream of the start codon. Each amino acid is brought to the ribosome by a specific transfer RNA molecule.
Yeah.
The type of amino acid is determined by the anticodon sequence of the transfer RNA. Complementary base pairing occurs between the codon of the messenger RNA and the anticodon of the transfer RNA. After the initiator transfer RNA molecule binds to the start codon, the large ribosomal subunit binds to form the translation complex, and initiation is complete.
Incision is complete.
In the large ribosomal subunit, there are 3 distinct regions called the E, P, and A sites. During elongation, individual amino acids are brought to the messenger RNA strand by a transfer RNA molecule through complementary base pairing of the codons and anticodons. Each anticodon of a transfer RNA molecule corresponds to a particular amino acid. A charged transfer RNA molecule binds to the
ये चार्जेड tRNA molecule
Yes, sir.
yemis pech chu lower amino
Amino
acid
acid.
woni godnikis pech kus amino acid chu
Methionine.
methionine बहुत अच्छा pat kım aysan emin zorat pat ayni timiyuş doyum av emis pech chu lower
Yes, sir.
ya क्या amino
Amino
acid
acid.
chu godnik kat site manza
Itra P side.
वो ये बाकी ये tim kas
Sorry,
manza
A side.
charged tRNA that means loaded with amino acid loaded with specific amino acid
Yes, sir.
loaded with
Specific amino acid.
The A site, and a peptide bond forms between its amino acid and the one attached to the transfer RNA molecule at the P site. The complex slides down one codon to the right, where the now uncharged transfer RNA molecule exits from the E site,
Yes, sir.
and the A site is open to accept the next transfer RNA molecule. Elongation will continue until a
when you ribosome pa kya hai
Yes, sir.
Hukun.
Opposite.
a kadam hukun a kodan hukun
Yes, sir.
mRNA pa kyu
Yes, sir.
mRNA pa yes direction mas upai yes tha whenever kas karta hai ribosome
A stop codon is reached. A release factor binds to the A site at a stop codon, and the polypeptide is released from the transfer RNA in the P site. The entire complex dissociates and can reassemble to begin the process again at initiation. The purpose of translation is to produce polypeptides quickly and accurately. After dissociation, the polypeptide may need to be modified before it is ready to function. Modifications take place in different organelles for different proteins. In order for a digestive enzyme to be secreted into the stomach or intestines, the polypeptide is translated into the endoplasmic reticulum, modified as it passes through the Golgi, then secreted using a vesicle through the plasma membrane of the cell into the lumen of the digestive tract. Proteins are needed for most physiological functions of the body.